anaerobic metabolism can be like in the microbiology realm, we tend to say anaerobic metabolism, but I think some other people might say more like anaerobic respiration. It means the same thing. So if you've heard it maybe one way or the other, don't get confused. It's basically the same thing. And so that's the idea where you have the electron transport chain cooking along. It's just that you don't have oxygen sitting at the end of it. Okay. And the end product is excreted. So you've already seen this. I just added the word respiration in case that helps you figure this system out. So if they're not using oxygen, last time we thought, okay, if they're not using oxygen and if they're a really savvy microbe, they're going to pick something close to oxygen on the redox tower. And so if iron is a choice, but it's usually for those that live in super acidic conditions, which is more rare than if you were to pick a nitrogen containing compound, which is more commonly found all over the place. So nitrate, NO3 minus, would be a really good option. And so a lot of microbes do that. They'll look around, they get to pyruvate through glycolysis, and they're like, where's my oxygen? And they're like, oh, bummer, I have no oxygen. So then they say, well, I'm not acidic. I don't care about iron in that case. I'll go with nitrate. That'd be my next best pick. And then you can see up here further, there's iron up here at a pH of 7. So don't get confused. It's just different habitats we're thinking about, okay? But anyways, nitrate is a good one. And so then we looked at that and how microbes can take it and put nitrate at the end of an electron transport chain. And then if they use that as a terminal electron acceptor, it goes to nitrate. And then like E. coli should just stop there. That's all it seems capable of doing. But like Pseudomonas can keep running a longer electron transport chain and keep going and going. And so we looked at here. So I wanted to point out that if you go from nitrate, NO3 minus, and then you start cooking along any point along the line, you start calling this process denitrification. And if you look up the definition, they usually say it's going from nitrate to all the way to dinitrogen. Okay, dinitrogen. And so I would like you to know denitrification. So the nitrification portion of that word, we usually think about nitrate. Nitrate's a very important compound to microbes as well as plants. I told you last time, plants love nitrate. So denitrification means you're taking nitrate and making it change all the way down to dinitrogen. And then if you just say nitrification, you mean you're making nitrate. And I'll show you that. I added this next slide in too, because sometimes people have had this maybe in environmental science classes or ecology classes. You might have seen this already. So what we were just talking about, the denitrification, is the idea of taking nitrate and taking it and converting it all the way to dinitrogen gas, which is up in the atmosphere. What we got, 80% in the atmosphere is dinitrogen. And then nitrification is the idea that you make nitrate, and we usually see that happening with ammonia. Ammonia is NH3, ammonium is NH4 plus. They're interconvertible, they go back and forth. Anyways, if you start with ammonia or ammonium and run a reaction to generate nitrate, then we say, oh, that's nitrification. And then we haven't talked about the other idea here. I don't know why I kept the circle, but and then if you take nitrogen gas from the atmosphere and fix it, nitrogen fixation. That is taking dinitrogen and converting it to ammonia. And this is very special. There's only a handful of microbes in the vast majority of microbes. There's really very few that can do this. And it's only microbes. Only microbes know how to do this. Take dinitrogen to ammonia. Fungi don't know how to do it. Plants would love to know how to do it, and they can't do it. But plants are smart because they partnered up with bacteria and said, I'll give you a home if you fix nitrogen for me. And that's what we see. So plants are smart that way. Yes. Pardon? Peas. Yeah. So sometimes they'll phrase it that way. Peas do nitrogen fixation. It's like, no. They partner with bacteria that do nitrogen fixation. So there's a lot of writing like that that's misleading. It's kind of, you can tell, a pet peeve with me because only bacteria can do that. And if you can discover or engineer a microbe or something beyond bacteria that can do this, that would be huge. Okay, so that's the nitrogen triangle. I put that in case some of you have had that in another class, if that's helpful. And then we looked at this last time. So here's your electron transport chain. On the left is aerobic respiration, where you have oxygen as the terminal electron acceptor. And then on the right is anaerobic metabolism or anaerobic respiration. All right, where you have nitrate sitting at the end. So that's pretty much what E. coli can do. But then, like I said, Pseudomonas can take it further and keep going. And in that process, they can still generate more proton pumping, which is good. And so you're going to test this tomorrow in lab, I believe. You're going to do the nitrate test with a couple of different microbes, and you're going to see what they can do, what they're capable of. It's a simple inoculation that you'll do tomorrow, and then you'll incubate and you'll work through some additions of chemicals next week to determine what they're doing. And all right, so I believe we left off here. So now this is a little different concept. So now we're not talking about what's the terminal electron acceptor so much. We're focused on what is kicking it off. What's the start of the electron transport chain process? Who is donating? What is donating electrons? So chemolitho, or just lithotrophy, is the idea that you have some kind of inorganic. So we can't do this. It'd be awesome if we could, but we cannot. And so we have to eat something that was once living, some organic material, to get our electron and energy sources. So there's plenty of options for microbes around the world. Just one volcanic eruption can give you a ton of hydrogen sulfide gas. So that's inorganic. And surprisingly, a lot of microbes like hydrogen sulfide gas, they strip electrons off of it, and it provides energy into the electron transport chain. Through the processes of mining and agriculture, we certainly give our environment quite a bit of inorganic sources of iron and nitrogen and others. In fact, they use microbes in mining industries in order to purify copper, for example. They're doing, you got to know microbiology in order to do that. Industrial wastes, of course, all kinds of wastes that we've provided into the environment, and microbes are actually like, yeah, I can use that. And it doesn't even poison them. It helps them. And then just through the action of other microbes, and let's say it's a soil environment, and you have some microbes that produce hydrogen sulfide gas, and then they release that. And then these other chemolithotrophs are like, hey, I can take that and I'll use it to strip electrons off. And we know a lot of microbes produce hydrogen gas, and some can produce ammonia. So you have these teams and consortiums of microbes that, saying one person's waste or trash is another person's treasure, that kind of thing. It's the same thing with microbes that all hang out together. They're all kind of doing their own thing in terms of their physiology, but it often works out where the waste that's getting excreted by one microbe is very happily taken by a nearby microbe and acted on for their physiology. So they're inorganic, and we know that all living organisms on the planet Earth are organic based, carbon based. So it's like, how are they doing that? Well, they actually take in carbon dioxide, and they're autotrophs in that way. So they can make their sugars, kind of like what plants do. And so they're just not using sunlight energy. They're using energy and electron coming from inorganic molecules. Some are funky. Some are like, yeah, I'll work on inorganic compounds. I'll strip electrons off of them, but they can't take CO2 and make sugars off of it. They're not quite that good. And so we say they're heterotrophs in that regard, like we are, like we have to eat something carbon-based. So if a microbe uses inorganics as an electron donor into the electron transport chain, but they can't use CO2, they have to eat something that was once alive, some carbon-based, then you call them mixotrophs, right? So this is a really odd group. Most lithotrophic organisms are autotrophic for sugar production. They can make their own. So really, like, sometimes you'll see categories lithoautotrophs because they're taking CO2 and creating sugars from that. All right, here's some examples. All right, so you can see there's lots of options depending on the microbe and where they live. So again, they're just assessing their environment that they've adapted to. And if there's plenty of hydrogen, they'll use that, keep electrons off of it. And what this is trying to get at is that this, at least a lot of microbes, again, if you can choose oxygen as your terminal electron acceptor, you're going to do that. And so what this is trying to show is that this is really an electron transfer chain process with oxygen sitting at the end. And they're not showing the free energy, but you get some nice proton pumping and then a proton gradient from these reaction pathways if you put it into an electron transport chain. So you can see it just depends what the inorganic is. Hydrogen is a favorite. We don't really like that, but there's all kinds of other options, iron and nitrogen and sulfur. All right, so there's lots of options, but basically all of these that are being shown, it's an electron transport chain process. So let me show you a picture. So hydrogen, H2, hydrogen gas is a very common strategy because there's a lot of H2 out there in many, many environments, produced by other microbes, for example, in particular. And so, and if you look at the redox tower, it's pretty high up on the redox tower, which means hydrogen gas is pretty good at giving off electrons. I use the analogy that I like to think about Tinkerbell with the pixie dust, and that I think about the dust coming off of her little wand as the electrons, and Tinkerbell is the hydrogen gas. Because it's just so happy to give off. Although Tinkerbell wasn't happy, was she? Wasn't she crabby? Wasn't she kind of grumpy? At least some versions of Peter Pan. Anyway, so I like to think of happy Tinkerbell. Maybe she was in an altered state and was happy. So anyways, what you see here again is this reaction that they've drawn, O2 plus H2. Of course, they're hiding all the fact that that's an electron transfer chain going on, where you have hydrogen getting stripped of electrons, the terminal electron acceptor is oxygen. All right, so let's just take a brief look at that. So I'm not gonna, ask you to memorize this from start to finish, but there's a couple of key things to see here. So I'm trying to prove to you that when you see something written like this, it's really, you got to think about this. It's an electron transport chain process where here's your hydrogen gas, and they're showing that it's getting stripped of electrons and getting passed through an electron transport chain process with these particular players. This microbe has some cytochrome complexes. And then you see oxygen as the terminal electron acceptor. And again, that's a smart move by the microbe because that gives you more bang for your buck. Technically, it gives you more proton pumping ability. So once you have a nice steep proton gradient, that's when you get the protons wanting to diffuse back through the laws of diffusion. And that's what's going to cause your ADP. They didn't even put the inorganic phosphate there. They should have put that inorganic phosphate, and then they react and get you your ATP. So this is lithotrophic because you're using that inorganic electron donor at the start of the process. So often with microbes living in the real world, if nitrate, NO3 minus, is going to get made, it's usually in a team effort. Usually what they see happening, especially a lot of the soil environments that they've sampled for this kind of thing, what you see is a team effort. So for example, the nitrosifiers, I've just given you a couple of genera names here. But basically nitrosifiers, what that means is they strip electrons off ammonia and you end up with nitrite. NO2 minus is nitrite. And then there's some other nearby microbes that go, hey, yeah, I'll take the nitrite and strip electrons off of that. And I'll make nitrate. And again, they don't care about the nitrate. They're like, that was just a game I had to do to get my ATP, do my redox balance, everything's good. And then they spit out the nitrate, but then of course, other microbes or plants will say, thank you for that, I'll use it. Okay, so I'm just trying to show here that you do often in a microbiology realm, you have teams or consortia of microbes doing this, their physiology, and they use the waste from one as their starting product on another microbe. I put this in too. I'll have this in the updated slides. If this visual helps you think about this idea of either making nitrate through the process of nitrification or taking nitrate and converting it to dinitrogen, then that's denitrification. So if that helps with what's going on here. I know when I was first learning those terms, it was confusing. So sometimes just seeing the visuals like this can help you lock it in. And then this is something you've probably not heard about in any ecology class. I don't think they usually don't talk about it in a standard ecology class, although I talk about it in microbial ecology. Here is Anammox. So it stands for anoxic ammonia oxidation. That's what they've abbreviated down to Anammox. So this is what they're talking about. They're taking ammonia and reacting it with nitrite. And so ammonia is going to strip off some electrons off of that. And in the process, you end up making dinitrogen. Well, not very many microbes can do this. If they can, though, it's a big payoff. So free energy of negative 357 kilojoules per mole. What do you need for ATP? Negative 32. So that's a lot of ATP you're getting from that one reaction. So they actually use this in the wastewater treatment plants in the cities. When your water gets flushed down the sink or toilet and then it goes to the wastewater treatment plant, part of the treatment plan is to have an area set aside to allow for this. Because ammonia, of course, is in the urine of, waste. And so you can easily feed a lot of those microbes, and they have those like standing pools where they create anoxic conditions and allow for the water to get treated. Just let the microbes do it. So they don't have to invest in some fancy technology to get rid of the ammonia. They just make the microbes happy and have them do it. And so it's a nice application of microbes, and it's pretty darn cheap as long as you know your microbiology and can keep them happy and keep them satisfied. So you have to make sure that it's an anoxic condition. It's also happening in oceans too. It's naturally. And it's part of the nitrogen cycle. When you think about dinitrogen in the atmosphere and then that getting fixed and then it's got to go back up and just go round and round and round. So we have a nitrogen cycle on planet Earth. They're really funky organisms when you look at them under the microscope. They have this internal membrane system called the anamoxisome. And that's all membrane, internal membrane system, where they stick all these enzymes and proteins in order to run this electron transport chain. So they're really busy. They have a lot of surface area in order to do this pretty fancy reaction. Okay, so I think you might have gone by wastewater treatment plants where they have pipes in the ground sticking out. They're usually fairly large pipes on the field above, like, well, it's more of a dump site, but wastewater treatment plants do it too, because of the production of methane. And so methanogenesis, pick that part, that word apart. Genesis is to create methane. Okay, so they're saying these are microbes that can create methane. And so what you have happening is they can take carbon dioxide or some other pretty simple carbon compounds and convert it to methane. Methane's a gas, it's a waste to them, so they'll just excrete it out. So far, the only ones that seem to know how to do this are the domain microbes that belong into the domain archaea. We haven't found any in the domain bacteria. So for example, they might do something like this. They'll take carbon dioxide and hydrogen gas, react it, and they'll get methane. Now again, they don't care about the methane. This is just a game to them where they live. They're like, I got plenty of CO2 around me. I have plenty of hydrogen gas. I have all the necessary proteins and enzymes and all of that, whatever I need to run an electron transport chain process and generate a proton gradient. And so that's what they're doing it for. And then they just get rid of the methane. Well, then there's nearby microbes that are methanotrophs. Troph means to use or to eat. And they're using or eating methane. So once the methanogens do their thing, create methane, get rid of it, then the methanotrophs nearby are like, hey, great, I'll do that. I'll take it. And then they run their own electron transfer chain process as well in order to get their proton gradient. So if you've heard that methane productions are part of global warming and climate change, and if they point the finger, what animal do they point the finger at for methane production? Cows, right? Cattle, cows. It's not technically them that's doing that. It's the microbes in them. Cattle don't produce methane. It's the microbes. And so they have a very special compartment as part of their digestive tract called the rumen. And it's animal digestive tract. So we point the finger at cattle because we raise so much cattle for food production and milk production that they're the primary culprit. If you just add up how many cattle versus sheep, elk, deer, and so on, or camels. Don't think about camels too much over here, but and so if you peer into that rumen in the digestive tract, which one of our former grad students did, they actually do these types of experiments. I think MSU still has this going on downstate where they do an operation on the cow. I think this is a dairy cow, and they create opening, surgical opening into the rumen, and then they like cap it off in between sampling. And then you can go uncap it and take a sample. So here's Dan taking a sample. You actually like put a sterile glove on up to your armpit and go in and take a sample. Apparently they keep the cows very happy. They're well fed and well taken care of, but it's like a long-term experiment where they can do these samplings over time and under different like dietary conditions, and they can track what's happening to the microbes in the rumen. So the rumen is chock full of billions upon billions of microbes, including the methanogens, but there's a bunch of others in there too. And it's essentially all anaerobic. They even have some eukaryotic type organisms that can handle being totally anaerobic. So really interesting experiments and things that they've learned about these very interesting pathogens and what they're doing. But you can see that even in humans, archaea can hang out in your intestinal tract. And but again, it's not the human producing methane, it's the microbe in an anaerobic environment. Even the hindgut of insects like termites. So you have some options for some interesting anaerobic environments where the methanogens have set up camp. So CO2 is a common idea, but there's others options, but pretty much they usually just say simple with like 1 or 2 carbon compounds, not really anything big, but basically it's this idea of an electron transport chain. They set up an electron transport chain using these substrates and do their electron transfer, and they link it to a proton gradient. And for a methanogen, they get a lot of energy out of that. So it's a good strategy they evolved. Has anyone done that sampling? Yeah. So do they literally, you just go in with your hand, or do they give you a tool and like, yeah. Yeah, yeah. But they don't like say, hold this like ladle and go in and just take a sample. Well, that's probably pretty rare. Not many people get to do that. So yeah. Okay. So let's do a Google form. Let's take a little bit of a break. I still have some more to tell you about physiology, but let's just do a short little question. I'm gonna pop, get out of this. No, I didn't test this beforehand, so hopefully it gives you a multiple choice question and then an essay question on a physiological reaction. Is that what it's giving you? I should have a reaction written out, and I'm asking you what type it is. So you can talk and use your notes. I want you to think through. You have to look at that reaction and try to figure out what it is. Getting close to wrapping it up. You don't have to go on and on and on for the essay portion. If you just give me one reason, that's fine. I don't believe it was set up to give you the answer right away, correct? So let's think about this. Which of the following best describes the reaction? We have a hexyl sugar, 6-carbon sugar, and you see the products are butyrate, acetate, hydrogen gas, and CO2. So could it be chemolithotrophic? No. Hexyl sugar means 6-carbon sugar, so that's not litho. Litho has to be inorganic. Can't be chemolitho. What do you think about the remaining options? Fermentation. So what's one reason why you would think fermentation? There's several reasons. Carbon dioxide showing up, acetate showing up. Both of those are really common in fermentation. So is hydrogen. Production of both. All three of these is really, really common strategy for a fermentation reaction. Is butyrate and acetate, it's not CO2, right? So it's a partial breakdown of a 6-carbon sugar. You only got one little CO2. If this had all been CO2 plus hydrogen gas, then it's like, well, that's not really fermentation. Fermentation is a partial breakdown of your starting sugar or molecule. You also don't see any oxygen in this equation. So aerobic respiration, not an option, which is on here anyways. Should have been, but I forgot to put it on there. Okay, so I'll look through some of your, I'll look through the essay questions later, but questions on this fermentation idea? Yes. Okay. Okay, we'll do another one like this probably Friday. Really quick here, let me, while we're taking a break, if you have your laptop, why don't you go into the EduCap page for lecture and I can get open the essay questions from the exam and we can go over that. Let's see. Let me just get it working here. Okay, if you refresh now and then go into essay questions, it should be showing you your answers. Is it? Hey, did I do the wrong one? Lecture exam 1, essay. All right. Now come on. All right. Now do a refresh. Now is it showing? What the heck? Oh, I know why. Never mind. I need more caffeine. All right. Lecture exam 1. Now let me. Okay. Now do a refresh. Okay. Sheesh. So look through your answers and what I've written typed up into the answer key. And then I'll talk to you a little bit after that. All right, so let's look at the question on specialized and then asking a little bit about generalized. So from start to finish, couple of things. Please be aware that a bacteriophage is not a prophage. So it's totally bacteriophage is the virus, okay? Bacteriophage, it's a virus or a phage that attacks bacteria. A prophage is a virus that is in the state of inserting its DNA into the host DNA, into the host chromosome. And then it just sits there in the chromosome. Then we say it's a prophage. It's pro meaning before. I mean, it's not quite an active phage. It's just kind of sitting there. Okay, so that was one thing I noticed confusion on. And then things that I was looking for is the virus, inserts its DNA. The DNA gets pasted into the host chromosome. That's the prophage state. With specialized transduction, what happens then is once the phage becomes active, instead of cutting out cleanly and just taking the phage DNA, it takes flanking DNA with it. I needed you to say flanking DNA with it or adjacent DNA. And then as it's packaging that DNA into viral particles, it's carrying along some genes from that first host. So then when it goes to the second host and pastes its DNA in, it's transferring genes from the first host to the second host. Usually the second host, if there's enough of the first host DNA, the virus can't actively create problems for the second host. However, the transfer of those genes could be an adaptive mechanism, useful genes. Also, the same exact virus can't seem to attack those. It gives the second host immunity, so to speak. The same virus won't attack them. So that's what I was looking for specialized transduction. And then the huge main difference with generalized is that you don't have this special section of the DNA that gets taken. It's all just random in general. All the host DNA starts to break into pieces and it's just some random bit that will go into the virus and get transferred to another bacteria nearby. Okay. And then most of you did really well on the endosymbiosis idea, but I did need you to clearly say that it was a eukaryotic, the evolution of the eukaryotic cell type. That was such a significant jump because before that time, it was only prokaryotic cell types. And then all of a sudden, boom, you get this eukaryotic cell type. That was a huge step in evolution. But most of you got nearly full or full points for that question. So great job on that. And the average on the essay was quite good for a first test, 75.5%. That's really good for essay, because essay, you got to pull it all out. It's not multiple choice. So if you have concerns about your multiple choice or the essay and want to go over it in more detail, I'm certainly happy to do that. We can go over it very often as we look at what you got right, what you got wrong. I can start to see a pattern, and then I can help advise you on, better strategies for the next exam. So please take advantage of that. I'm very happy to help you. Once you get a learning pattern down that works for you, it's amazing how much that helps. No. The next exam's all just over new material. Yeah, the final exam will be partly cumulative. So the next exam is week 6. Is that next week already? So I'm thinking not this Friday, but next Friday again is probably what we'll do. And it'll be on physiology, the stuff that we're talking about. So if you've been sitting here the last couple lectures going, ew, yikes, then please come and see me and go through things. Because I think I did mention that when we started the physiology, when the second exam in this class is a lot of physiology, and traditionally, historically, that's what I see the lowest averages of all the exams.
Will we be able to respond to multiple choice questions?
If you come in and set up a time with me, we'll go through that. Yeah.
Cool. Can we do it on EduCount ourselves?
No.
Oh, okay.
I have it set up so that you need to come in with me and we'll go over it. So please, for this next exam coming up, like I said, it's probably not this Friday, but next Friday, and definitely heavy on the phys. Sorry, but I figured it's better to rip the Band-Aid off and do all the phys in one stretch. and then we're good. All right. I'll see you on Friday. Valentine's Day.
Will you one of those exam for us before the cumulative exam? So it'd be nice to be able to see
Yeah,
what we got.
I know what you mean.